Good morning, one and all present over here. I, Shruti Patel, Assistant Professor from Parul Institute of Pharmacy, hereby welcome you at webinar platform of Parul University. It's a great pleasure for me to introduce our today's speaker, Mr. Hardik Joshi, who is going to deliver a talk with us about development of roadmap for generic sterile drug product. He is currently working as a manager at Sun Pharma since last 3 years. He is having 9 years of experience in a food and drug department. He has already worked senior executive at Sun Pharma and as a trainee in F&D department at Zydus Cadila, Ahmedabad. Herewith, I would like to request Mr. Hardik Joshi sir to share a knowledge on a generic sterile drug product. Over to you, sir.
Yeah, thank you, Shruti, providing for an introduction, but a little bit the modification in the introduction is required. So let me take this opportunity to introduce myself one more time. So yeah, as myself, Hardik Joshi, I did my B Pharm and M Pharm from LM College of Pharmacy, and I completed my PhD from the Kadi Sarva Vishwavidyalaya. Currently, I am associated with Amneal Pharmaceuticals since last 10 years and mainly taking care about complex injectable and ophthalmic product. Prior to the association with Amneal, I was associated with Zydus Cadila for 4.5 years, and before that, I was associated with Astron Research Limited, which is the R&D of Intas Pharmaceuticals, right? So this is the brief about my, academic and the professional experience. Right. So is my screen is visible, Dr. Asha?
Yes.
Is there any modification with respect to enlargement is required, or it's okay? Is it fine, right?
Sir, you may continue. Your slide is visible.
Yeah, okay. Thank you. So before moving to the actual talk regarding this development roadmap for the generic sterile drug product, I just want to, I'm curious, like, to understand, like, how many participants are here for this talk, Dr. Asha? Okay, fine. No issue. I think you're on mute, Dr. Asha. So, what is my understanding that the participants are mainly from Bachelor of Pharmacy, then Master of Pharmacy, and some of students maybe from PharmD or the PhD, right? Okay. So
yes, majority PG and PhD scholars.
Okay, great, good to know. Fine. So, before moving to the actual talk, again, I would like to thank you for providing me an opportunity to deliver a talk where, I can exchange the industrial experience and the knowledge to, academic students. I am sure this talk will really help to, understand, to gain more insight how this generic industry working, right? So, before moving to the actual talk, just I would like to give the little bit background about this topic. What is the idea, for selecting this particular topic? So in the next 1 hour talk, we are mainly going to talk about development roadmap for the generic sterile drug product, right? So we are going to talk about what are the different milestones for the development activity, how this generic industry work, what are their specific aim, what are the different milestones, and what is the actually sterile drug product, and how we used to work in our day-to-day life, right? So in this entire topic, we are mainly going to cover the different aspect. So we are not going to talk about any specific technology, any specific doses form, any specific the process in detail. But what we are going to talk, like how this industry is working, what is the sterile drug product, how we used to submit the ANDA, what are the different cross-functional department are there, how we used to coordinate with different the discipline in order to make a successful submission of ANDA, and what is going to happen after submission of ANDA, and how we used to commercialize our products. So these are the entire process, and we'll talk about start to end, like starting from the introduction of any new product and the commercialization. Of course, any generic industry is always working for the revenue purpose. So how we use to do the commercialization of any product from R&D to the market. So we are going to will discuss in detail about this entire process. And as I understood, like the most of the audience are from the masters and the postgraduate students, so they might be thinking that they will pursue their career in maybe academic, maybe industry, and maybe some of they will do their own business. So I think this kind of this presentation will give a lot of insight, lot of the knowledge how we used to work in generic industry. So that will really help you to choose your career path in different discipline, right? So I'm sure this is going to be very informative, very knowledgeable to all of you, and I would suggest like just note it down the few of words, a few of the sentence which I'm going to talk, and you may Google it, and will do the more the research on that particular topic. So that will give a lot of insight, because we are not going to cover a lot of things here, but yeah, we'll touch the some aspect over here. And if you have any questions, you can raise your hand at any point of time, but I would suggest you can ask the questions at the end of this session, so the flow of this presentation will not distract at any point of time. All right, I think does it make sense, right? Okay, fine. So let's get started on the actual on the topic, that is the development roadmap for the generic drug product. So as you see in the title, we are going to talk about the development roadmap. We are going to talk about generic industry, and obviously we're going to talk about the sterile drug product. Just checking that my slides are moving or not. I think the slides are not moving, right? Okay. Is it?
Yes.
Okay, fine. Thank you. So as I specifically mentioned that we are going to talk about sterile drug products. So what are the different sterile drug product that we used to work? So the one is injection. So the injection, I'm sure like you all know, like the solution which is available in pre-filled syringe, or the solution which is available in glass vial, or like liquid solution which is available in infusion bag, right? So these are the injectable sterile drug product. Sometimes in case of the injection, your doses form may get changed. Maybe that is suspension means the insoluble the particle, but still we are going to inject as a IV route. So that is again the part of the sterile drug product. Maybe like we are working with emulsion doses form, depending upon the therapeutic category, depending on therapeutic need. Yes, we are also working on the microsphere, which is a particular drug delivery systems. We are also working on the liposomes, and again the ophthalmic drug product. So what is the most common in all the doses form? It should be sterile, because we are dealing with the IV administration, or we are dealing with the topical, specifically ophthalmic. The main criteria for all this drug product, it should be sterile, right? So that's why we are calling a sterile drug product. So being a formulation scientist, being the R&D scientist, we used to work in the different dosage form. Just keeping in mind that all should be in the sterile nature, right? So that makes this dosage form little bit different from the other dosage form. Now let us talk about like what are the cross-functional department are there. So when we work in the generic industry, we used to work with different department, right? We need to coordinate with different departments. So being a formulation scientist, we need support from the different department in order to make our program successful, right? The program over here is generic development, right? ANDA submission. This is our main aim. So we need to develop a product and we need to submit. But for all this activity, we need support from the different cross-functional department, right? So what are different cross-functional department? Let's start with the business development. So what is mean the role of business development department is they will identify the opportunity, right? Depending upon the market scenario, they will identify the opportunity where company will get more and more revenue, right? So this team will identify and then they will introduce the product in the system, right? So this is the main work or responsibility, I would say, for the business development: identification of opportunity and then introduction into the system at the right time, right? Then analytical development. So again, the analytical development will work upon the different methods for the analysis purpose, right? Different method, I would say, like HPLC, UPLC, GC, then HRMS. So different the methods. Then packaging development department, yes, they will identify the right packaging for this particular the product. Let's say if it is injectable, then right selection of vial, rubber stopper, PFH. So that's their key role. Then sourcing and the purchase. So definitely this team will help us to identify the source where we can get our active, where we can get our packing material, from where we can get our raw material. So by that we can start our formulation development activity. So we need their help also to do the formulation development activity. Obviously production. So production is again where we need to execute our patches at a higher scale. In R&D we used to work with 1 liter, 2 liter, max to max 5 liter, but in case of the production we may end up with 30 liter, 50 liter, 100 liter, maybe 500 liters. So the production where we'll do the actual manufacturing and from where actual commercialization of the product will happen. So this is the the production. QC, again, like when the analytical development people will work upon the method, then this will do the testing. So transfer and the testing will the main responsibility of this QC. So they will release your API, they will release your the excipient, they will release your packing material, and finally they will do the stability testing of your ANDA batches. So again, their support is really important. QA, obviously they need to take care about the quality, because at the end your the production facility is going to be inspected by the different regulatory authorities. So QA role is to make sure that we are well within the established quality system. So that's their role. So that we need their support. Obviously the regulatory affair, because regulatory affair is the main, I would say, the bridge between the formulation development and different department to the agency. So they used to prepare the lot of document. They used to review lot of document as per the requirement, and then the collectively they will submit the ENDA. So ENDA is it's a the collection of a lot of document that has to be submitted to the agency so that they can review what we did, right? And IP team, obviously. So as we are working on the generic product, we are going to copy of innovator. So our main aim, what is the generic industry? We are just doing a copy from the innovator. So what innovator used to do, they used to file some patent. So we need to take care that patent also. We should not infringe that patent. So our IP team will help to give the direction with respect to the patent. Patent may be of process, patent may be of the formulation, patent may be of any polymorphic form of API. So they will direct us like what specific API we need to select, what specific process we need to select, so that will not end up with infringement of that particular patent. So in a generic industry, these are the different cross-functional department where any M.Pharm, any PhD or a postdoc student can pursue their career, right? So depending upon their interest, they can build their career in IP, in regulatory, maybe in sourcing, maybe packaging development, analytical development, and yes, the core is the formulation development department, I would say. So these are the different opportunity where where you can think upon to building your career. And yes, these are all the different department where formulation development team has to coordinate in the day-to-day activity. I think this give a lot of insight about how we as a R&D we used to work in the generic industry, right? So moving to the next, let us talk about what are the common challenges or issues associated with the sterile drug product. So as I mentioned that what are the major quality attribute of the sterile drug product? It's sterility, right? And again, being this liquid product, there are a lot of the things we need to take care during development activity. What are the different things we need to care during development? Q1, Q2. So we'll talk about what is Q1. We'll talk about what is Q2 in subsequent slides. So we'll have idea. So you can note it down this particular, the word that Q1, Q2, right? So we used to talk regularly about Q1, Q2 in our day-to-day, the work life. So we'll talk more about what is Q1, what is Q2. Again, being this liquid product, again the depending upon the doses form, the process development is again very critical, right? So let's say if you're working on the liquid injectable, their process is a little bit simple. But when you move on to the suspension, when you move about to the emulsion, microsphere, liposome, the process development is very, very important, and it's going to be create a lot of the challenges for us, not only at R&D state but also at the manufacturing scales, because as we change the scale, again the challenges are different. So that is the key for the development perspective. Obviously we need to take care about the stability, because our main aim is to develop the product, to develop the process in a such way that it will give the chemically and the microbials stable product, right? So again, this is very important for us to to look at about the stability. Scale up, as I mentioned that as we change the scale, the challenges are something different. So what sort of the I would say the facility we have. So we need to forecast that we need XYZ for doing the scale up, for doing the manufacturing at that scale. So that also we need to take care during our development activity. So let's say being a scientist, you develop your product at R&D scale, but you will not able to scale up at your facility, then it is not useful. So being a scientist, we need to take care that my product should be scalable, my product should be manufactured at this manufacturing scale. All right, so that also we need to take care. Container closure, yes, why container closure? Because in case of the sterile drug product, these products are going to be come directly in contact with your container closure, so that will define the stability of your product. So right selection of container closure is also important for the sterile drug product, right? So let's say you take care about development, stability, and scale up, but you have not selected your container closure properly, then again you end up with stability issue, right? So it's equally important that you should select right container closure. And last but not the least is the GMP compliance, right? Being the sterile and drug product, there are a lot of challenges are associated with the GMP compliance, right? Because these products are directly, this product is going to directly administered into the blood, right? The sterility is main, but how to maintain the sterility at a manufacturing site, that is very key, right? So that's I mentioned that the QA and the production are going to create vital role, in order to maintain the GMP compliance. And again, FDA, I would say, or I would say like any regulatory, the body is going to come to your site to inspect that, whatever you did, that you did properly or not, you're maintaining your compliance or not. So they are also going to check. And if they raise any questions, then again, then there is a big question on your facility, and again, you're not going to sell your product, you're not going to do the commercialization, and again that will end up with revenue issue with any organization. So being a sterile drug product, GMP compliance is a very the important, right? So this is overview about like what are the challenges associated with the sterile drug product, what are the key aspect we need to take care about, any development activity of sterile drug product, right? Right. So moving to the next, let's talk about this NDA versus ANDA versus 505(b)(2), right? So these are very important terms. I am sure like few of heard about these terms, and for few this is the new term. But let's talk about what is NDA, ANDA, and 505(b)(2), right? So as I mentioned that we're talking about the generic industry. So our main aim is to develop ANDA, that is abbreviated new drug applications, right? See, again, that is generic drug application, I would say. So what we are going to do, we are going to copy NDA, new drug applications, right? So our main focus is to copy NDA, being a ANDA applications. So in ANDA, there is no requirement of preclinical, that is animal study, or a clinical or a human study in order to establish the safety and efficacy. So definitely, your development time is going to be shorter. You will end up with low cost, because moment you do the clinical study, so a lot of patients, lot of facilities are going to involve, that increase your cost of your development, right? So in this case, in ANDA, in order to establish safety and efficacy, no preclinical and clinical studies are required. But yes, if your dosage form is suspension, your dosage form is based on the particulate matter drug delivery systems like microsphere, like liposome, right, where you need to prove the bioequivalent. So your formulation should be bioequivalent to that of RLD, right? That is NDA product. We'll talk more about what is bioequivalent, what is the definition of bioequivalent. But this is overview of about ANDA. So what is ANDA? Abbreviated new drug application, that is generic applications. Now let's talk about NDA. So what is NDA? It's new drug applications. So like new chemical entity which should be safe and effective. So whenever this innovator company identify new chemical entity, they need to establish the safety and the efficacy of that particular drug as well as the formulation. So in order to prove that the safety and efficacy, they need to do the lot of animal study, they need to do a lot of clinical study in order to come up with good API and with good formulations, right? So in order to conduct this preclinical study and the clinical study, it requires a lot of time, lot of energy, lot of manpower, and again, lot of money. So that's the reason this will end up with high cost. And again, because of this particular process, as they need to generate a lot of data, they need to generate a lot of studies, and the costs are involved, that particular molecule, that particular formulations, as you see, the cost of the RLD is always high, right? So the innovator company are selling their product on the premium price, right? So whenever they introduce new product, right, if this is the only product, so they will charge at a higher price. Let's say maybe for one injection, they may charge for $500, they may charge for the $1,000, depending upon the therapeutic category, right? So all this depends upon the what amount of cost they incur during establishment of the safety and efficacy of that particular molecule, right? And in case of ANDA, being a generic product, we are charging little, not a little, but very less for our product. Why? Because the development cost is very low, right? And the time is also very low. So we cannot charge like, innovator. Let's say I am also going to sell my product at $500. No, you can't. Why? Because you will not able to justify, the price, right? Because you end up with very low cost. So you need to put your cost very reasonable. So that's the reason. That's the basic difference between, NDA applications and the ANDA applications. So I'm sure like you have a fair enough idea about what is NDA and what is ANDA, right? Now let's talk about 505(b)(2). So what is 505(b)(2)? Again it is a new drug application only, right? But what I would say, it is in between NDA and the ANDA, right? So again, in case of 505(b)(2), the small modification in the approved drug product is required, right? Let's say my NDA is lyophilized product, right? If I will convert lyophilized to the liquid formulation, then it would fall in the 505(b)(2) category, right? Let's say my formulation having leuprolide. Let's say if I will formulate my product with leuprolide acetate, that means I'm changing the form of API. It will fall into the 505 category. And as the safety and the efficacy is already proven by the NDA application, so you need not to do the many major preclinical or the clinical study, right? So again, the costs are low, but these are the somewhat, I would say, like the innovation with respect to the existing formulations. So again, this is the 505(b)(2). It's not NDA, it's not ANDA, it's in between the NDA and ANDA, right? So this is clear-cut difference between NDA, ANDA, and 505(b)(2) applications, right? I'm sure like you have like a fair idea about all these three terminology, right? You can Google it, you will get more insight. Most of all the literatures are available in the open forum. You can see like what kind of modification will fall in 505(b)(2), right? So this is NDA, ANDA, and 505(b)(2). Being a generic industry, our area of interest is ANDA, right? Okay. Now let's talk about therapeutic equivalence, right? So as we are working with ANDA product, as we are working with a generic product, this is the term where we need to, we need to understand. So to understand the therapeutic equivalence, let us understand what is pharmaceutical equivalent. So what is pharmaceutical equivalent? Same amount of active ingredient in same dosage form, right? So same amount means, let's say if I am working for any injectable product where my active is paracetamol, right? So for that RLD product or NDA product, the concentration of active is 10 mg per ml, right? And it is in a liquid injection form. So if I am working with ANDA, I have to use same active ingredient, that is paracetamol. I have to use same doses form, that is liquid injection. And I have to use same concentration, that is 10 mg per ml. If I will comply with this, then my product is pharmaceutical equivalent with RLD product. Got it? Simple. This is innovator. This is my product. My product should have same concentration, same active ingredient, and same dosage form. Then it is pharmaceutical equivalent, right? Simple definition. Then what is bioequivalent? Comparable bioavailability. That means rate and extent of adsorption should be same. Let's say I'm a copy of this RLD, right? So when I administer my product, it should have same rate and extent of the adsorption. Then I can claim my product is bioequivalent with innovator product. If I able to meet this criteria of pharmaceutical equivalent and the bioequivalent, my product is therapeutic equivalent, right? Let's say I'm using same amount of active ingredient, same ingredient, and same dosage form, and it has same rate and the extent of absorption. That means it is going to be therapeutic equivalent. Let's say if I am targeting for any muscle relaxation, the therapeutic activity, that means yes, it is going to provide same therapeutic efficacy, right? So if your product is pharmaceutical and bioequivalent, that is sure that it will going to deliver same therapeutic activity, right? So being a generic application, you need to understand this word very important: pharmaceutical equivalent, bioequivalent, and we'll combine these two, it will make therapeutic equivalent, right? Okay. So these are again the important terminology that we should have the idea. Okay. Now let's talk about para patent certification, right? So as I mentioned that different cross-functional department are working. So one of department is IP, intellectual property, right? So this department will take care about the patent part, right? So whenever we are submitting any ANDA, that ANDA has to be qualified for any of para 1, 2, 3, and 4, right? Now let us understand what is para 1, para 2, para 3, and para 4, right? So paragraph 1 is where patent not submitted. Let's say I know I'm developing paracetamol injection, right? I'm going to submit my ANDA, right? So I need to check whether any patents are there, whether patents have been expired. If patents are there, when it is going to expire, right? And let's say if I'm working on the paracetamol injection, there are no patents submitted by the innovator company. That means I am falling in paragraph 1 ANDA certification, right? Now, what is para 2? Patent has expired. That means, if I am developing any product, let's say today it's April 22, right? For that particular paracetamol injection, patent is already expired in 2020, right? And if I would like to submit my ANDA in 22, that will fall in para 2 category, correct? Now let's talk about para 3. That means patent will expire, right? Let's say I am right now I am in April 22. I am working on a paracetamol injection. For this particular product, patent is going to expire in 23, right? For that, the ANDA will be para 3 certification, right? So I will say that I will sell my product, or I will commercialize my product only after expiration of the patent. So that disclosure we have to upfront mention in our ANDA applications, right? So you need to tell to the agency that yes, we know that patent is 23, we are submitting this, our application is 22, but we will commercialize our product after expiration of the patent only. So that is para 3 certifications. Yeah. Now let's talk about para 4. It's very important. Being a generic industry, we need to understand about para 4 certifications. What is para 4? Patent is invalid or will not be infringed, right? Now let's say when we are developing paracetamol injection, we checked in Orange Book, right? It says about patent is going to expire in 2030, 8 years after, right? So what is going to happen? if you submit your application, if you get the approval, but you will not able to commercialize because patent is still 2030, right? So being generic industry, what we used to do, we thoroughly the review the what the patent has been submitted by the agency. Let's say the patent is mainly for the formulation. So what as a scientist we used to do, let us evaluate different options where we can bypass this patent, right? So we will not infringe this patent, right? So being a formulation scientist and being a IP team, so we used to close do the coordination with IP team. Let's say for the paracetamol injection, we come up with some solution, where we modify concentration, where we modify the excipient which has been claimed in the patent. So do we really infringe? So what formulation scientists we used to do, we propose some sort of the formulation to the IP team, and IP team based on their expertise they will review and they say yes, this is out of patent, let us move ahead, right? So this is the para 4 certification where we as a generic industry claim that yes, we are not infringing this patent. This patent is invalid based upon the what the formulation we have developed, right? But just keep in mind, this is really very, very expensive affairs, right? What happened that you based on your close coordination, based on your close discussion with your IP team as an organization, you decided yes, let us move with a para 4 certification. But what is going to happen, innovator can sue you, right? They claim that no, your formulation is infringing in our patent. Then again there is a 30-month stay, right? So your product cannot be approved till 30 months, right? Again you need to hire a lawyer. Then again there is a conversation between lawyer. Again we need to involve the cost for the lawyer, right? So the para 4 is a very risky affair where you know to be very, very cautious if you are developing para 4 formulations, right? So these are the different patent certifications where being a generic industry we need to work upon, where we need to do the close coordination with our IP team, where a lot of legal the formalities are there, so we need to understand what para 1, para 2, para 3 and para 4. So what is the safest is para 2 and para 3, right? And the riskiest is para 4, correct? So 1, patent not submitted, 2, patent expired, 3, patent will expire, and 4, patent is invalid and will not be infringed, right? So again, this is very important terminology we need to understand, when we are working in the any generic applications. Okay. Now next, move on to the So being a formulation scientist, what we used to do, right? I already mentioned that we need to coordinate with different department, but there are certain, or I would say the major, the aspects or activity we need to do as a formulation scientist, right? So now let's talk about what we used to do as a formulation scientist. We used to do the literature review, right? So any product has been introduced by the business development, right? Then what is our first role? Do the literature survey. We need to understand what is this product, what is the primary packing material, what is the formulation, what is the therapeutic indications, is it Q1, Q2, right? So that will come to know by referring a PIL. What is PIL? Product information leaflet that is readily available from the drugs at FDA site, right? So you need to just type paracetamol, you will come to know this is the PIL, right? Then again from the drugs at FDA site, we need to go through the summary basis of approval, that is SBOA. So what is SBOA? There are a lot of document that has to be submitted by innovator company, right? About their product, about their different studies, right? There are a lot of administrative documents are there. So that will give a lot of insight that what has been done by the innovator company. So some of the open information they used to share, but some of the critical information we will not able to get it from this SBOA because they will hide, in this particular document. So we'll get some sort of idea, right? Yes, what different kind of the study they have done, and that will also give the some insight that how we used to, strategize our formulation. Then patent search, I already mentioned that is there any patent or not? If a patent, what are different patent? What are the type of the patent? Is it formulation? Is it API? Is it a process? So we used to do the patent search through the Orange Book patent, right? Again, we need to check the bio guidance, right? So let's say if we are working with liquid injectable, which is available in a solution form, in that case, there is no bio studies required, right? But if you are working on the particulate matter doses from, let's say, suspension, let's say the emulsion, let's say the microsphere, liposome, right? Where we need to prove that my product is bioequivalent with RLD, right? That is bioequivalent, right? For that, uh, you know, FDA has published bio guidance, product specific bio guidance, where FDA will tell that you need to conduct this kind of the bio study in order to prove your product is bioequivalent with RLD, right? Then again, research article, obviously, I'm sure, like, being a, you know, uh, M.Pharm student, you're working on the different project. Being the PhD student, you're working different projects. So you used to do the literature survey. You used to download a lot of research article. You used to read that. So similarly, we are also doing same thing, so that we'll understand like what has been done for this product. What are the different stability related issues? So we will come to know through the different research articles. And based on this literature survey, we what we used to do, we used to define the development strategies. How we will develop this product. What would be the regulatory strategy? What would be the IP strategy? Right? Let's say if there is a patent, then what is our strategy? How we should work? If it is regular, if it is a liquid, if it is a microsphere, then what is a regulatory pathway? How we should submit this product? What are the different prerequisites with respect to the submission of the product? Right, that regulatory people will tell us, right? And the being a formulation, we define the formulation. How I will develop my formulation? How will make the stable? How will I select my container closure system? Right, so that we will do during very initial phase, very moment the product introduced. So that is the literature review. So this is again very important. We used to take a lot of time for doing literature search, literature review, so that at initial phase of development will have lot of insight about the product, right? So very important step, that is literature review. Okay, now let's talk about the product similarity, right? I already mentioned in initial slide that Q1, Q2, and Q3, right? So now let us understand what is Q1, what is Q2, what is Q3. So this is the common the terminology we used to talk during our routing uh, activity, right? Now, what is Q1? Q means qualitative, right? Qualitative similarity, right? I will show you one example, so you will understand more about Q1. Now let's talk about Q2. So whenever we are saying Q2, that means quantitative, means the concentration, I would say, right? And Q3, that means structural similarity, right? So this is Q1, Q2, and Q3, qualitative, quantitative, and structural. So whenever we are developing any generic product, any generic applications, your formulation should be Q1, your formulation should be Q2, and your formulation should be Q3, right? Now let's take an example so that you will have more idea about what is Q1, Q2, and Q3. Right? See, let's assume that we are working on the acetaminophen injection, right? So for acetaminophen injection, what is Q1? So Q1 is this particular the column: acetaminophen, mannitol, cysteine, then diabasic sodium phosphate, sodium hydroxide, hydrochloric acid, and water for injection. So this is qualitative, right? So that is Q1. Now what is Q2? That is quantitative. Means for acetaminophen injection, I am going to add acetaminophen at a concentration of 10 mg per ml. I am going to add mannitol at a 38.5 mg per ml. I am going to add cysteine hydrochloride monohydrate at 0.25 mg per ml. So that is quantitative, right? Qualitative, quantitative, and then Q3, that is structural similarity. So when you develop your product, right, it should have same pH with RLD when you compare. It should have same osmolarity when you compare with your innovator product. If you are working on the suspension product, the particle size should be same. Particle shape should be same. Surface area of particle should be same. Polymorphism should be same. It should match with your dissolution. It should match with your viscosity and the sedimentation rate. So Q3 will vary depending upon type of dosage form. Let's say if you're working on the emulsion, right? So in case of emulsion, globule size is your Q3, right? So depending upon the type of dosage form you are working for any sterile drug product, your Q3 will be vary, right? So this is Q1 qualitative, Q2 quantitative, and Q3, that is structural similarity, right? So I'm sure like you will have idea Q1, Q2, and Q3, correct? Okay. Now let's talk about QBD. So again, this is very important concept that is quality by design, which is systematic approach for the development, right? So this is quality by design. So whenever you're working on any sterile drug product, we used to develop product by keeping quality by design concept in mind, right? So what are the different arm of quality by design? One is QTPP. We'll talk more about what is QTPP. CQA, right? The quality, critical quality attribute, right? In case of QTPP, it's a quality target profile, right? Risk assessment, then development of robust process, and then control strategy, right? So this is so whenever we used to develop our product, quality by design concept should be in mind, so that our product should be robust, our process should be robust, and based on the development we used to define the control strategy, so that that will deliver consistent product over a period of time, right? Now let's talk about QTPP, right? So what are the different quality target product profile? Let's say we are working on the acetaminophen injection, right? So these are the different QTPP element: your dosage form, your route of administration, your dosage strength, stability, drug product quality attribute, container closure, and administrative information. So these are the different QTPP element for the acetaminophen injections, and for each element there is a target, and for that there is some adequate justification, right? Why we have selected injectable solution? Because we are claiming our product as a pharmaceutical equivalent. We talked about, right, about pharmaceutical equivalent, right? Why is this route of administration? Because innovator is also claiming intravenous, so I am also claiming intravenous administration. innovator is claiming 10 mg per ml, so I am also claiming 10 mg per ml. So that makes my product a pharmaceutical equivalent, right? Obviously, stability, right? Always we are targeting 24 months shelf life for particular storage conditions, right? So equivalent or better than the RLD. That different the CQA. We'll talk about the CQA. So this is the one of the element of quality by design. So we need to take care about this QTPP during our development activity, right? Now let's talk about the CMA, CQA, and the CPP, right? What is CMA? Critical material attribute, right? Let's say we are working on the acetaminophen. So the assay of acetaminophen, LOD of acetaminophen, and then residual solvent of acetaminophen. It's a critical material attribute. Let's say we are working on the acetaminophen injection. We are adding excipient. So their attribute also we need to take care about development. How this CMA will impact on the quality of your formulation, that you need to assess, right? So it is equally important you should know about critical material attribute of your API, your active, and your excipient which you are going to use for the formulation purpose, right? Then CQA, critical quality attribute. First is critical material attribute, second is critical quality attribute, right? So what are the different CQA for acetaminophen injection? Description, pH, percentage transmission, absorbance, assay, related compound, particulate matter, sterility, and bacterial endotoxin. So these are the quality checks, I would say, or I would say like critical quality attribute that we need to keep in mind during development of acetaminophen injections. So again, I would like to simplify about this CQA. So what we used to do, let's say I have prepared one acetaminophen injection batch with some process, right? I have the prepared bulk, I have filter, and then I have filled into the, let's say in 50 ml glass vial, right? So after preparation of this particular batch, you have to taste that batch with respect to this CQA, right? So what we used to do, being a formulation scientist, we prepare batch, we have submitted to our counterpart, that is analytical development. So moment you submit the sample to them, so they will start analyzing sample with respect to different CQA like description. They will test for the pH. They will test for the assay. Let's say you are claiming that this particular batch has a concentration of 10 mg per ml. Then they will test and they will let you know, yes, that is 10 mg per ml or 100% of your assay. So these are the different CQA where our analytical and the QC will let you know about your the product. And last is a CPP, that is critical process parameter, right? So whenever we are working on the different doses form, again the CPP will change. Let's say you're working on the simple solution. So what is your CPP? What is your critical process parameter? That is mixing. So you need to ensure that when you manufacture a batch, your acetaminophen should dissolve. So you need to take care that in what time it is going to dissolve. Let's say 5 minutes, 10 minutes, 15 minutes, so that it will give the clear solution, right? It should be clear when you add acetaminophen, because we are working on the clear solution. So that is your CPP. Now let's say you are working on the suspension product. Again, your CPP will be entirely different, right? In case of your suspension, mixing time of API is not your CPP, correct? Why? Why? Because in suspension, the API is not going to be solubilized. API will remain in insoluble state. So in that case, your CPP is not mixing time, right? So in case of your CPP, the particle size reduction process is become your CPP, right? What is the pressure? What is the time required to reduce the particle size or to hit the particle size in line with RLD, right? That become your the CPP. If you change again doses from like emulsion, the globule size. So in order to meet your globule size, there will be a different critical process parameter, right? So moment you change the doses form, your CPP will be different, your CQ will be different, and your CMA will be different. Let's say you're working with a particular method delivery systems like microsphere. So in that case, you are going to add polymer, right? Different polymer depending upon the the microsphere. So polymer attributes we need to take care or we need to assess during the development. How the change in the polymer characteristics will impact on the our CQA, that you need to assess, right? So the meaning to say, this CMA, CQA, and the CPP will vary depending upon the type of your dosage form. Right now we are talking about the simple liquid injection, that is acetaminophen injection. So these are the my CMA, CQ, and the CPP. Right now, let's talk about what are the different lab studies we used to do during the formulation study. So yes, feasibility trial, because we don't have any idea about this product. So we used to do how this product is going to manufacture, how it is going to behave, right? Then obviously prototype formulation development. Then we used to do the stability study as per the ICH guideline. Again, process optimization, because we used to do the optimization with respect to the different scale. Let's say for 1 liter the process will be different, maybe 5 liter the process will be different, and again if we go up to the 30 liter, 50 liter, so that process also we need to optimize depending upon the scale. Different development studies like compatibility, bulk hold, photo stability, thermal study, diluent compatibility, filter validation study, d-value study if your product is TS. We'll talk about what is TS product in subsequent slide. Then freeze drying microscopy for the lyophilized product which is available in cake, right? Then obviously analytical method development and the method validation. So these are the different lab study we used to do being the R&D scientist. These are the common study. Again, type of the study, the duration of the study will be different depending upon your doses form. But these are the common, the critical development study we used to do for any you know uh, generic product development, right? Now let us also understand the stability study, right? Let's talk about the injectable product as per the ICH guideline Q1A. So this guideline is readily available on the open forum. So you may download and you can read. But this is the grips of the stability study for the any injectable product. Let's say you're working on the acetaminophen injection, right? And you have referred the peel of acetaminophen injection where they are claiming that product can be stored at 20 to 25 degrees centigrade, right? That means by referring the peel, let's say your the storage condition of your product is 20 to 25, that is room temperature. Then what will be your storage condition during your stability study? It's 40 degree as the accelerated, 30 degree for intermediate, and 25 degree for the long term, right? If the storage condition is room temperature, then accelerated stability condition is 40 degree, intermediate is 30, long term is at 25, because your storage condition is room temperature. Let's say your product has a storage condition of refrigerator, that is 2 to 8 degrees centigrade, right? Then during stability study, the accelerated condition is 25 degrees centigrade and long term is again 2 to 8, right? And let's say your product has a storage condition is minus 20, then only one condition, that is long term. So during your development study, during your ENDA submission batches, the batches has to be charged for the stability studies, and you need to generate the lot of data with respect to the different stability conditions. Accelerated means at the 3 month or a 6 month, how your product is going to behave, right? So you need to generate a lot of data, accelerated, intermediate, and long term. Based on this data, you can submit your ANDA. Right? Now, let's talk about ophthalmic product. Right? There is a slight difference with respect to the stability condition when we talk about ophthalmic product. Right? Let's say ofloxacin ophthalmic solutions, which is having a storage condition of room temperature. Right? So what would be the accelerated stability condition? Yeah, 40 degree, but there is a change in the humidity. Right? In case of the ophthalmic, as it is stored in a semi-permeable container closure system, the humidity condition is little bit different, right? In case of long term, 25, yeah, temperature is same, 25 degree, but humidity is 40 degrees, 40 plus or minus 5% of relative humidity, right? See, in case of injectable, 40 degree and 75% RH, right? But in case of ophthalmic, not more than 25%, right? In case of injectable, long term, 25 and 60, right? But in case of ophthalmic, 25 and 40. So these are the storage condition, I would say the stability conditions already mentioned in ICH guidelines. So you can refer ICH guideline, that will give more insight why there is a difference in the humidity conditions, right? So why I'm talking about the stability? Because we need to prove that my product is stable. How you can prove your product is stable? By generating stability data at accelerated and the long-term conditions over a period of time, like 3 months, 6 months, 9 months, 12 months, 18 months, and 24 months. Let's say your product comply to the specification of long-term storage condition for 24 months. That means the shelf life of 24 months can be granted for your product, right? So in order to establish the shelf life of your generic product, the stability study is very, very important, right? So that's the reason I have captured the stability study slides over here, so that you will understand, why it is important, right? So not even you need to do the stability study at R&D stage, but when you manufacture the exhibit batches at manufacturing site, for that batches also we need to conduct the stability studies, right? So R&D stage also you need to do the stability study, and at the manufacturing scale you have to conduct the stability studies. And based on all this data, we can submit our product at a 6-month stability, and we can get the shelf life depending upon the data at long-term storage condition, right? So it is equally important to understand this stability study and their conditions, correct? Okay. Now let us also understand, the process development part. So I talked in a first slide that the common challenges or issues associated with the sterile drug products. So for one of these, the process development, right? So when we are talking about sterile drug product, there are two processes where we can manufacture our product. One is aseptic and second one is terminal sterilization, right? So aseptic process will apply to the thermal sensitive product, right? So whenever your product is thermal sensitive, means if you increase the temperature, you are going to get the impurities. Yeah, impurity will develop significantly. That means your product is thermal sensitive. So what is your process? The filtration through 0.2 micron filter is your process. See, I told, like, what is important the quality attribute for the sterile dosage form is sterility, right? So for thermal sensitive, you have to filter through 0.2 micron in order to make your product sterile, right? For that, you need to do the media fill simulation study. Now, again, the media fill or a process simulation is an entirely different chapter where production and the quality will come in picture, right? But being like the graduate or a postgraduate student, you should know, yes, there is a media field requirement with respect to the aseptic process development, right? And as you are doing with filtration, yes, you are adopting a filtration, you need to do the filter validation step. Again, filter validation is entirely different subject. We can talk about 1 or 2 hour on only on filter validation, only on the media fill process. But these are the very important terms with respect to the process development of the sterile product with respect to the aseptic process, right? Now let's talk about the terminal sterilization process. So where in this case your product is thermally stable. That means if you increase the temperature up to 121 degrees centigrade, you are not going to get any impurity. Your product is rock stable. No worry with respect to the change in description. No worry with respect to the impurity profile. Right. So you can do the autoclaving or you can do the steam sterilization of your finished product. Finished product is the product available in vial, product available in its final doses form, product in infusion, product in PFS. So you can do the steam sterilization, right? For that, media fill simulation is not required. So that is again difference. Again, as you are doing terminal sterilization, again this is the CPP, is critical process parameter, where you need to optimize and validate the cycle, and for that you require D value. Again, TS cycle development and the validation and the D value is an entirely different subject. We can talk 1 hour, 2 hour on this subject, but we should know there is a D value requirement, there is a optimization and validation is required. Media fill simulation is not required. And what is autoclaving temperature? It's 121 degrees centigrade for 15 minutes, right? So this is the aseptic versus terminal sterilization process difference. Okay. Now let's also talk about the packaging, right? So as I mentioned that your product is going to come in directly contact with your packing, and that packing is also define the stability of your product. So it's equally important that you should select the right primary packing material. You should select the primary packing material in a such way that it will give the highest stability of your formulation, right? So again, there is a category of your packing material. Primary, that means primary means which is in contact with your product. That is primary. Secondary, like carton, right? So that vial with rubber stopper and seal, you are going to put in another carton. So that carton is your secondary, right? And tertiary, that means the 50 carton, again you are going to pack in some other tertiary pack, right? Other protector which will create the ease during the transportation. So that is a tertiary packing. So our area of interest is only mainly primary and secondary. And again, from primary versus secondary, main area of interest is primary, because